Many people think that the research of cancer is a more recent development, but actually it dates back all the way to ancient Egypt. The first record we have of cancer is from around 3000 BC in the Edwin Smith Papyrus. Also, one of the earliest people to do significant cancer research would be Hippocrates. He lived in 400 BC and he was known as the father of medicine. He coined the term carcinos or carcinomas in relation to tumors. And this is thought to be because of the finger-like projections that emanate from tumors and the fact that carcinos is the Greek word for crab. We now know that these finger-like projections is the vast vascular system that a tumor needs in order to develop. And so we're going to be talking about that, and we're also going to be talking about other factors that make an environment perfect for cancer development, for these cancer cells to grow. So we'll be talking about how our environment affects cancer growth and how inflammation and the immune system affect that. And then we'll also be talking about our genetics and what factor genetics have in the proliferation of cancer. And then we're going to touch a little bit on the tumor microenvironment and what needs to be there in order for cancer cells to grow. Our environment plays a huge role in whether we will develop cancer or not. It is said that 30% of cancer-causing factors are out of our control, which means that 70% of factors that cause cancer are within our control to change. The first time that a cancer-causing agent was correlated to the workplace was in 1775 when Percival Pott discovered that chimney sweeps had a higher risk of developing squamous cell carcinoma of the scrotum. And this wasn't just correlated to chimney sweeps. This is also correlated to any man working with distillates of coal or mineral oil. And this is because all of these things contain polycyclic aromatic hydrocarbons. And when we are exposed to those compounds, our body metabolically activates them, and then they are able to bind with DNA and cause cancer. And depending on which polycyclic aromatic hydrocarbon you are exposed to is what cancer will form. The many different forms of cancer form because of these. And this was the first time that a hazardous workplace environment was discovered to cause cancer, but it definitely wasn't the last. In the 1800s, it was the aniline dye industry, and those employees were forming bladder cancer. And then in the early 1900s, a bunch of people were hired to paint with cadmium paint, and they all developed bone cancer. A lot of them developed bone cancer. And other scientific things that were happening around this time would be the discovery of X-rays by Nikola Tesla in 1896. And then in 1898, Marie and Pierre Curie discovered radium and polonium. A few years later, in 1903, radiation was used for the first time to treat cancer. It was used to treat basal cell carcinoma, and it actually cured these patients. But it wasn't until the 1940s that we realized that radiation could be a bad thing, and it could actually cause cancer in certain forms and in certain strengths. And then all of this stuff happened, and many people in industries were dying because of what they were exposed to at the workplace. So in the 1970s, the United States put together rules and regulations for workplaces in order to protect their employees. And part of this is testing different compounds to make sure that to see if they are carcinogenic or not. And part of the way that we test these carcinogenic compounds would be using different forms of assays. And so we have an Ames assay that will test for genetic mutations, and we have the HPRT assay, which uses human somatic cells to test for carcinogens. And then we have a few different ones that look at chromosomes. We have the chromosomal aberration assay that looks for abnormalities in metaphase chromosomes. We have the sister chromatid assay, which will look at duplicated sister chromatids. And then the other one that looks at chromosomes would be the micronucleus assay, and that looks at the number of chromosome breaks. And then the last one that will look for carcinogens would be the COMET assay, which uses fluorescence microscopy to look for DNA breaks. But it's not just enough to identify these carcinogens. We also have to have a way to test people to see if they have been exposed to carcinogens. We are all exposed to them to some extent in our daily lives. It's just the threshold of what is too much so that cancer will form. In order to test people for carcinogen exposure, we use biomarkers. And there are 3 different categories of biomarkers. We have biomarkers of effect, which test for specific carcinogens or their metabolites. And then we have biomarkers of exposure, which will test for certain biological changes that occur after somebody has been exposed to a carcinogen, but before cancer has had a chance to form. And then the last category of biomarker would be biomarkers of susceptibility. And these will test for certain genetic makeups that will put somebody at a higher risk of developing cancer. The workplace isn't the only place where we can come into contact with carcinogens. One of the most common carcinogens is tobacco smoke, and we were able to correlate tobacco smoke and lung cancer in the 1950s. And it isn't just a one-time exposure to this, it is long-term exposure. So somebody who has been smoking for many years or has been exposed to smoke for many years. And this is the same case with the chimney soot and scrotal cancer. These men were exposed to this soot day after day after day, and it wasn't until 15 to 20 years down the line that they actually developed cancer. Well, tobacco smoke causes cancer because there are many known carcinogens in the smoke of tobacco that cause DNA damage, and they are able to alter cellular growth. Our diet plays a huge factor in whether we will develop cancer or not. 14 to 30% of cancer deaths are related to being overweight or obese. And this is in many different forms of cancer, not just one specific type of cancer. The chemical compounds in food and the overall consumption of food both play a role in how our diet affects cancer formation. It has been found in animal models that when the overall consumption of food is reduced, so is the risk of cancer. And this isn't fully understood, but it's thought to be because of the different compounds, like inflammatory compounds, that our body releases when we overeat. But I think that one of the biggest things that people think of when they think of food causing cancer would be the actual chemical compounds in our foods. And this can be chemical compounds that already exist in the food, or it can be compounds that are added via pesticides and things like that. So fruits and vegetables would be associated with things like antioxidants and other good compounds that actually reduce your risk of cancer. However, there are foods like sugars and saturated fats and other foods that create an inflammatory response in our bodies, and that inflammation, if continued over time, can be a good environment for cancer to form. Inflammatory responses are both natural and necessary for our bodies to survive. And an inflammatory response is our body's immune system fighting off pathogens. And some of the cells that are used in the immune system are mast cells, and they are able to secrete cytokines and chemokines, which cause inflammation. Another group, which is the smallest group of white blood cells, are eosinophils, and they are used in parasite infections and allergic diseases. Neutrophils are used mostly in acute inflammation, and they are able to secrete reactive oxygen species to kill off pathogens. Dendritic cells are another form of immune cells, and they are able to stimulate naive T cell proliferation in order to stimulate the immune system. Macrophages are used both in acute and chronic inflammation, but they are found in higher quantities in a chronic inflammatory response. Natural killer cells, or NK cells, are what are used to fight off tumors and viruses. And then the last type of immune cells that we'll talk about will be T lymphocytes. And T lymphocytes are divided into two groups, CD4 plus and CD8 plus. We've talked about the immune system and inflammatory responses and how those play a role in cancer. But now we need to talk about what stimulates the immune system, and that would be viruses and bacteria. It was in 1911 that Peyton Rouse made the first correlation between a virus and cancer. And he did this with what is now known as the Rouse sarcoma virus. And his studies were done on chickens, so he would inject healthy chickens with this virus, and it was found that tumors would form at the injection site. Other viruses that are associated with cancer would be the Epstein-Barr virus, and this is a double-stranded DNA virus that causes Hodgkin's lymphoma. Probably the most well-known virus to cause cancer would be the human papillomavirus, or HPV, and this is found in cervical cancers. It causes other forms of cancer, but the most well-known would be cervical cancer. And there are high-risk strains and low-risk strains of HPV. HPV 6 and HPV 11 are two low-risk strains that can cause abnormal pap smear results and genital warts, but they will not cause cancer. HPV 16 and HPV 18 are high-risk strains, and they are found in 99% of cervical cancer diagnoses. There are two proteins, E6 and E7, that are associated with the high-risk strains of HPV. And these proteins are able to cause chromosomal destabilization and therefore cause cancer. Viruses aren't the only infectious agents that are able to cause cancer. Bacteria also play a role in cancer formation. H. pylori was found in 1982 to cause gastric ulcers, but later on we also found that it can be an issue with causing gastric cancers. We talked a lot about things in our environment that we can change because they cause cancer, but now we're going to talk about something that we can't change, our genetics. In the 1970s and 80s, a big topic of cancer research was somatic mutations. And somatic mutations are mutations that occur naturally in the somatic tissues over somebody's lifetime, and these can alter genes. In the 1970s, Varmus and Bishop were studying the Rouse sarcoma virus, and initially they were studying the replication of the virus. But after 1974, they started to research the mechanism by which the Rouse sarcoma virus changes cells. And in this research, they were able to discover the SRC oncogene. Now, an oncogene is a gene that can cause cancer. They also discovered proto-oncogenes, which don't have the ability to cause cancer unless they are mutated. So they found this cellular SRC gene, which is the proto-oncogene, and the viral SRC gene, which is the oncogene. Now, there are a few more oncogenes that we spoke about very commonly this semester. One of those would be the RAS oncogene family, and that was found with the rodent sarcoma virus. And then the other oncogene that we spoke about a lot was the MYC oncogene, and that was found in avian myelocytomatosis. We've talked about oncogenes, which are cancer-causing genes, and now we're going to talk about tumor suppressor genes, which are genes that stop the formation of cancer. There are multiple different models of tumorigenesis. One of those is the multi-step model of tumorigenesis, and this says that there are multiple different intermediate steps that a normal cell must go through before turning into a tumor cell. And this model suggests that a tumor cell needs 2 or more oncogenes in order to become a tumor cell. But when we started testing tumor cells, we found that they either contained RAS or MYC, but not both of the oncogenes at the same time generally. Most of them only contained one or the other. There's another model of tumorigenesis that says that a carcinogen will get into a target tissue and will cause a proto-oncogene to become an oncogene, and then that oncogene will cause tumorigenesis, will cause a tumor to form. In the 1970s, Alfred Knudsen did studies on familial retinoblastoma, And what he found in these studies were that people that got familial retinoblastoma only had one mutation, whereas sporadic retinoblastoma patients had two mutations. So this showed that there was something genetic in the family line that was causing people to be at a higher risk of forming retinoblastoma, because all they needed was one mutation to cause this cancer to form, whereas other people needed to have two mutations, and that was less likely to happen.
Our discussion of tumor suppressor genes wouldn't be complete if I didn't mention one specific tumor suppressor gene, the p53 gene. Mutations of the p53 gene is one of the most common abnormalities found in human tumors. Normally, the p53 gene works as a transcription factor, and it is activated in response to many different things, including DNA damage, activated oncogenes, and telomere shortening. And normally, when it is activated, it would induce apoptosis or cellular senescence in cells. However, if it is mutated, the p53 gene can cause tumorigenesis.
Angiogenesis, or the development of a vascular system to a tumor, is necessary for the tumor to grow. And this correlation between tumorigenesis and angiogenesis was first put together in 1971 by Judah Folkman. And we know that a tumor needs this vascular system to grow, but it can hurt us or it can help us. If it has a good vascular system, that means we can get drugs to the tumor more efficiently to kill it. But that also means that it's a higher risk tumor and it's able to grow better itself because of this vascular system. Now, our vascular system that we have in our body is very highly organized, but the vascular system of a tumor is very disorganized. And this is thought of to be because of either oncogene activation or tumor suppressor gene inactivation. And this disorganization causes the blood flow to the tumor to be very uneven. Now, we do have treatments that we use that are anti-angiogenic treatments, so they're able to stop this vasculature from growing. And that is able to help us kill the tumor and to create a better environment for the tumor to not be able to grow as well. While vasculature creates a great environment for tumors to grow, so do wounds. And we found this out from the studies of Rous sarcoma virus. And this was found out through the injection of the Rous sarcoma virus into chickens. And the only place that a tumor would grow was the site of initial injection. And this was thought to occur because the wound that was inflicted on the chicken when the needle was placed into its skin to inject the virus. About today, we know that cancer has been talked about and researched for thousands of years now. The first record we have of cancer research was in 3000 BC in an ancient Egyptian textbook. And then we moved to around 400 BC with Hippocrates, and he's the father of medicine, and he coined the term carcinoma in description of a tumor. Then jump forward to the year 1775, and that is when we first correlate occupational health hazards with cancer formation. And we have these environmental factors that cause cancer, but then in 1863 we are able to correlate inflammation with cancer through the research of Virchow. In 1889, shortly thereafter, Paget comes up with the seed and soil hypothesis, which says that these cancer cells will move into other tissues, and if the tissue is suitable for growth, then another tumor will grow. Then we have 1903, the first radiation treatment, which was able to treat basal cell carcinoma. In 1911, we correlate viruses with cancer through the Rous sarcoma virus, and the studies he did with chickens. And then in 1945, we have our first death from radiation. And this was really when we realized that yes, radiation was good and has been used to treat cancer since 1903, but 1945 hits, we have this death, and we realize radiation can cause cancer. In 1950, we found a correlation between smoking and cancer formation. So 90% of lung cancer deaths are actually found that smoking was a factor in that. In the 1970s, we have a lot of research going on. We have Judah Folkman talking about tumorigenesis and angiogenesis and how those have a huge correlation with each other. We have Alfred Knudsen talking about researching familial retinoblastoma and the mutations that occur in there. And then we have Varmus and Bishop, which discover oncogenes, which is a huge factor in whether cancer will form or not. And then in 1982, we have the correlation between H. pylori and stomach ulcers. So this fact will later go on to let us know that H. pylori also causes gastric cancer. And then finally, we have in 1984 the discovery of our high-risk HPV viruses, HPV 16 and 18. So we see through all of this research that our environment and our immune system and inflammatory responses and also our genetics and genetic mutations all play a role in whether we will develop cancer or not. And it's not just one of these things that causes cancer. It's a combination of all of these things and the environment that you are in that can cause cancer. And we've known about cancer for thousands of years now, but it's only within the past 100 years that we've really done a lot of research and moved forward a lot in both treatments with cancer and the causation of cancer.